Why?
Limitations of the card.
Oh no.
It said card speed, not space, so maybe it just means that we're just going slow.
Okay, okay.
It's like trying to catch up with itself, so it's not.
You're gonna be like, that's gonna be a weird video.
Right, yeah. So there have been many studies before ours that have looked into gene expression in tomatoes. So where we kind of stand apart from the rest of the crowd is that most everybody else are comparing mutant lines and wild type. And that's not very applicable to agriculture. So we're specifically looking at agricultural lines, which I'll talk about a little bit later. But these are just a couple of graphs that I found. So the one on the left has is a massive RNA-seq study that just kind of looked at where the different genes are expressed in the tomato, but this has no no correlation to lycopene. So we have no idea how much lycopene is in each of these tomatoes. We just know where the genes are being expressed. That's not very helpful for us. So and then the one on the right just compares two different wild type and mutant lines, which again, not that applicable to us. And then another problem that we might possibly run into is with chromoplasts. So chromoplasts develop from chloroplasts, which chromoplasts are pigment storing organelles for the tomato. They sequester carotenoids, lycopene included, into these chromoplasts. And the thing is that several high pigment cultivars of tomatoes have been found to have either many extra chromoplasts or just large compartment chromoplasts. So this means that their genes might not necessarily be upregulated like other cultivars are, but they might just have extra space for the lycopene to reside in, and meaning that they would produce more lycopene, and they do. So what we're doing now is looking at breeding lines, like I talked about, 50 different breeding lines, but they're from all over. Like we're trying to get kind of a broad look at the gene expression from many different breeding lines from around the world. So America, Italy, Japan, Spain, Israel, France, lots of different places. And so the goal is really to correlate how these lines that are used in agriculture or will be used in agriculture, some of them haven't been released yet, but some of them, but the ones that will end up in agriculture eventually, how those cultivars specifically use their genes to produce high lycopene. And so we are going to correlate between gene expression and lycopene concentration. I'll talk about that a little bit more in a second, with the goal of eventually being able to modify the plants in order to increase based on what we're seeing in the high lycopene lines. So this is what we have currently been doing. We have 3 biological replicates, 50 different lines, and they're currently in a greenhouse in Kannapolis. Many trips every week. So we're looking at 6 growth stages, which you can see on the left. So I'll point to them. So we have immature green, mature green are these 4. We have breaker, orange, pink, and ripe red at the top. So these are the 6 different growth stages. So I forgot to include a picture, but we are flash freezing them in liquid nitrogen. So we pick them in Kannapolis, bring them back, cut them open, which you can see at the bottom, talk about that in a second, and then throw them into liquid nitrogen, grind them, put them into -80, and then we'll extract the RNA later. So as far as the tissues that we're looking at specifically, we're specifically looking at pericarp. So most other studies that have looked at gene expression in tomatoes just look at pericarp. So we're trying to kind of follow that trend. So we just kind of cut a slice and throw it in and then grind it up and move on. So there's about 900 samples. So what we're doing is